Okay, so measles can become a chronic continuous infection. So ones that you might know better, HIV, hepatitis B and C, but essentially they're constantly detectable in the blood. Symptoms are present, and that infection is chronic. So it's lasting for long, long periods of time. So it's always there. So when I mention measles, it's interesting that it can be an acute infection, but unfortunately, it can remain in the body and stay latent for a long time, so up to 7 to 10 years. Um, Generally speaking, then this is one of the big reasons why we should get the measles vaccine. I never had measles. I don't have to worry about carrying that measles virus, post infection. But our smallest, most vulnerable individuals in the population, our children, are at high risk if their initial infection occurs before age 2. And your book states that before age 15 months. So there's 4 stages you can see. All of them essentially are causing a loss of motor functioning, a loss eventually of brainstem functioning, and death. So it's really important to know that this can be what should I say, a condition that some children who have measles might have. So it's really important to early diagnose them. Oops. All right. Oncoviruses. So if you've ever heard that the human papillomavirus, the HPV, causes cervical cancer, then you've heard of this idea that there are viruses that cause cancer. So we have a vaccine for that. Both male and female teenagers are getting that vaccine so we can eradicate cervical cancer. So anytime a woman has had an abnormal pap smear, she may have to redo that, and then eventually they would be testing to see if those cells were positive for HPV. So experts estimate 20% of cancers are actually caused by viruses, which to me in my mind seems terrifying. But then I say, okay, like we've known this, we're treating this, it's not like some great surprise. But definitely the research that we need to do to ensure that we're protecting ourselves through modern medicine is important. So we essentially see that there's what we call the transformation. This is the effect of those cancer-causing viruses on host cells. So again, here's a really great figure. That virus on the far left side is being taken up by that cell. And this happens to be a retrovirus. So it has RNA and is making DNA. And the DNA that it is making is full of these what we call oncogenes, or genes that cause cancer. Now there's various ways that the virus can cause cancer from the genes. In this one, it says it's leading to uncontrolled cell growth. So usually when we're thinking about tumors, we fall into like 2 categories, malignant and benign. So benign, just like its name kind of sounds like, oh, that's benign, oh, that's nothing, don't worry. Essentially, that kind of tumor is cells that are dividing unregulated and causing a growth, if you will, or a mass, but that mass is not metastatic. That mass is not breaking up and, like traveling through the bloodstream or lymph nodes to cause problems somewhere else. So a benign tumor is treatable. Malignant tumors are the ones we're concerned about, right? Make sure you have it excised, get all the margins of that tumor and so on, and then it hasn't spread. In the center, we see other retroviruses. So again, the viral RNA now is going to make its own DNA. So the retro there. And we see that the host cell is going to take up that oncogene and it's going to be replicated with the host cell. And then essentially it's going to make its own proteins. So it's making these oncogenic proteins. Far right-hand side, DNA tumor viruses. So here we have one of those icosahedral viruses being taken up. The viral DNA is in the cell, and it's going to directly produce proteins that lead to uncontrolled cell growth. So it's this uncontrolled cell growth, which is really kind of like a definition of cancer. Okay. All right, let's see. So once the cell has been transformed, we see that that host cell is going to demonstrate increased rates of growth. There will be changes in their chromosomes or changes in the cell surface molecules. And you can see it has this capacity to divide indefinitely, which means there's no tumor regulation. In eukaryotes, we have tumor suppressor genes, and these genes are going to look for unregulated cell growth or look for mutations that would cause that. So that's a super important protective mechanism that eukaryotic cells have. So again, we talked about HPV, that's the human papillomaviruses. We see that there's 3 other viruses there that are known to cause cancer in mammals. Okay, so how do we cultivate and identify animal viruses? Well, it's kind of cool, but essentially, I'm going to move on from this slide. We can use actual host animals. So specifically bred, we have a picture of a white mouse there. We can use rats, hamsters, guinea pigs, rabbits. Sometimes we use invertebrates. So you see a caterpillar there on the lower left-hand side. And sometimes we use bacteriophages. Remember how bacteriophages are viruses that infect bacteria. So we can use any of those to study essentially viruses. We can also use bird embryos. So that could be a variety of birds. But essentially, if you think about a bird egg, is it this nice enclosed, very well protected and contained environment? It's got the shell and it's got a few layers inside that shell. And essentially what we can do is take the viruses and we can culture them or grow them essentially in that, it's actually a fertilized egg. So we can replicate those viruses within the egg. And so you can see there's just different areas. You can do the amniotic cavity, we can do it in the yolk sac or the chorioalonic membrane. You're not injecting it directly into that bird fetus, okay, or embryo. So essentially we can grow viruses in this way. So you might have heard of vaccines, like if you have an albumin allergy or an egg allergy, there's the way that they make some viruses would be using some egg protein, not viruses, vaccines. So you would not be able to have that particular vaccine if you have severe egg allergies. So here we see it, we're injecting the virus into the egg. Again, it can go into those 3 different areas. Then we can isolate, purify, and inactivate the virus. So we call this an attenuated virus. But essentially, when we purify that virus, we can use it for vaccines. So there's a little bit more to this process, but there's lots of different kinds of viruses, and one of those is, oh no, not attenuated. Attenuated means it's still partially kind of alive. This one would be completely inactivated. Okay, so we can also grow and study viruses in little tiny sterile chambers. So you would take some cells of interest. So they show us in the picture on the lower left-hand side that essentially we're taking some lung tissue. And once you know like all the requirements of lung tissue, you can grow it in these little culture vials on the right-hand side. So essentially what happens is you grow the cells and you want like a single sheet of cells. And that is very conducive to viral multiplication or division. So essentially we would introduce a virus of interest and see whether it infected the cells. Now, how do we tell whether it infected it or not? Essentially, we're looking for what we call plaques or big areas where cells are absent, okay? So that's this whole area here. It looks like somebody just kind of erased them. But essentially, when that host cell becomes infected, it's going to forfeit itself, of course, because its genetic machinery has been hijacked by the virus. And we see that the virus is released by that host cell and it radiates out and it kills other host cells. So eventually you see these whole areas where there are no cells. So this gives you an idea that, yes, indeed, those cells were infected by that virus. And we call those like areas of clearness, we call it a plaque, okay, because it's responding to the areas of dead cells. Okay, so that's it for our second part of microbial nutrition and growth. And next we'll go on to microbial genetics. As always, let me know how I can help.